Hello everyone, my name is Kiah Ferrell. I am an environmental health researcher. Today I will be presenting to you all about gene-environment interactions. I will also discuss the disease that could be caused by a gene-environment interaction. Here is a list of the topics that I'll go over. Before I begin to talk to each of you, you all must understand the difference between a gene and the environment, and also understand how genes can trigger complications such as health issues. First, I will discuss the definition of a gene. A gene is the basic unit of life. Genes consist of chromosomes. Chromosomes are structures in a gene that are arrayed in 23 pairs. You get one chromosome from your mom and one chromosome from your dad. The X chromosome is from your mom and the Y chromosome is from your dad. DNA is considered your genetic makeup. Genes are composed of DNA. There are four base pairs, which are adenine, thymine, cytosine, and guanine. And for the gene regulation side, now genes can be turned off or turned on. This may sound a little difficult, but it's quite simple. Basically, compare the situation of a light switch. For example, say your house was considered the cell. So you walked into your house and the light was on, but the rest of the lights were off. Well, when each cell expresses, for example, turns on, it is the fraction of the gene that's only turned on. The rest of the genes, in this example, the rest of the house, are turned off. These factors also play key roles with genes. What is a gene expression? A gene expression is the process in which the information from a gene is made into a gene product. The expression of a gene in an organism can be influenced by external and internal factors. Gene expression is turned off rather than turned on simply because a complex of DNA, which is also known as chromatin, and proteins known as histones are found inside the nucleus. DNA methylation is basically the silenced gene expression and is associated with histones. Epigenetics is the study of the changes that occur by gene expression rather than the genetics itself. It is possible for cells to maintain the same histone code and DNA methylation patterns through cell divisions and can lead to epigenetics, and some changes to epigenetic can lead to diseases. Imprinting is the silence of one of the two alleles in the gene for the cell's entire lifespan. An allele is a gene that is made from mutation. The epigenetic marks takes place on the genes during reproduction, which are copied into each cell, and this affects genes over a time frame. Genes are also silenced by X inactivation. This means in females, one of the two copies of the X chromosomes are shut off and compacted. What is an environment? The environment can be related to anything depending on the situation. It could be the air that we breathe, walking in a department store, the neighborhood we live, or the workplace, and so on. But for the purpose of this presentation, it is to inform you on how the environment affects our bodies. The environment can be within our bodies, and the environment can be outside our bodies. Within our bodies will be like the cells and the genes, and outside our bodies will be like the air pollution or any other nature aspects that can affect the way our bodies function and react. What is a gene-environment interaction? Most diseases result from many interactions with genes and environmental factors. Gene-environment interactions are affected by certain environment exposures by disease risk in people who have other genotypes. Genotype is your complete genetic identity, basically. Gene environment interactions are influenced by the environment through external and internal factors. With external factors, such as the world a person grows up and lives, for internal factors would be like the hormones within the body and the cell divisions. An example of an external factor would be temperatures, chemicals, drugs, and etc. And this can turn genes on or off, as I previously stated. And this results into a way a person grows up. An internal example would be the factor that affects gene expression. Here I have a list of some environmental agents that could affect the human body. Differences in gene factors can lead to some individuals responding differently when they have been exposed to the same environment agent. Furthermore, some individuals have a lower risk for diseases through the environment, while others that are likely to get them. Now that you understand the difference between gene and the environment and the definition of gene environment interactions, I now then will move on to my disease of topic. The disease that I will be discussing is autism. There has been evidence shown that it is linked to gene environment interactions. According to an article on autism and genetics, autism tends to run in families. Identical twins are likely to both have autism because they share the same genes, rather than fraternal twins. Autism is pretty common. If one child has it, then the risk of the second child is about 2 to 6%. Now, if autism is due to one single gene, then the likelihood of the child getting it is about 25 to 50%. Basically, in simpler terms, there are a lot of genes involved with autism. Some scientists have discovered that 20 out of 23 chromosomes have regions that may be important to autism. What exactly is autism? Can anyone answer? Well, autism is a multiplex neurobehavioral disease. Multiplex means that it consists of many elements. In this case, it will be problems such as social interactions, languages, and speech skills. Furthermore, autism is a disorder that affects the way the brain reacts to different abnormalities. I feel like this slide is the most important slide of my presentation. The real question is, how is autism considered a gene environment disease? For the first bullet point, it states here that high heritability is seen in autism. Well, basically, you know now that the term gene is the basic unit of life, which in more scientific terms, gene is the basic unit of heritability, for instance. For genetic factors, they include genetic epidemiology and biological pathways. Genetic epidemiology is the study of genetic factors with environmental factors. This basically is what I was talking about when I said about the twins and the likelihood of they can get autism. For biological pathways, this deals with genes and structures within the body. Genes such as NLGN3 or NLGN4X and also SHANK3. These three genes could have problems resulting to autism. Now I would recommend you all that you go look up these three genes and get a better understanding of them. Now, it is very important that you know that there's much more environmental factors rather than exposures and chemicals. But autism risk factors can also include influences on a parent's age, wellness and nutrition, and what infections a woman has while she's pregnant. For environmental factors, such as the immune dysfunction in transcriptome states as here. Immune dysfunction simply means that there are problems in the immune system of the body. There has been studies shown that there is an increase in activated, and what activated basically means that it's turned on. So there has been increase in turned on cells, basically. Transcriptome basically deals with the gene expression for the brain of an autistic child and a normal child. Basically, they're just trying to compare the two brains to see what exactly is the problem. For epigenetic dysregulation, you should understand epigenetics by now. But basically here is the abnormalities happen with the expression of many genes without harming the DNA sequence. DNA methylation, histone methylation can be discovered through gene mutation or environmental exposures. There are a few elements associated with autism, such as mutation in genes involved with epigenetic regulation, and also chromosome regions that result to parental imprinting that contribute to autism. Last but not least, prenatal and perinatal. Now these words are kind of similar, but you can understand that pre going to mean before and peri is going to mean after. And they're basically meaning before birth and after birth. These factors can also contribute to autism. Such infections that the mother may have or any problems happening while giving birth can trigger autism with these two factors. Now this concludes my presentation. But we all must understand the differences between gene and the environment to better understand the concept of gene-environment interactions. The causes of autism aren't really understood, but there has been studies that link them to gene-environment interactions. For example, if a mother is exposed to some environmental agents, she could pass it on to her child. Hopefully this presentation was beneficial to you all, and now that you can be more aware of gene-environment interactions. These are my references. Are there any questions? If not, thank you.